This video will be on Chapter 8. This is the second part on chromosome variation. So previously we talked about aneuploidy and polyploidy. In this video we will talk about smaller scale chromosome rearrangements such as deletions, inversions, translocations. So here's a summary of the types of chromosome rearrangements we're going to talk about. This is a duplication. You have this small region, the E and F region, now is found twice in this chromosome. In a deletion, you have a small region of the chromosome that is missing. And so here you've lost this E and F region. In an inversion, you switch it around. And so even though you don't lose any genetic material here, nevertheless, you change the location on the chromosome where these particular genes are located. Finally, in a translocation, a segment of one chromosome will move onto a different chromosome. First, I'd like to talk about duplications and deletions. Most of the time, this happens because of a mistake in crossing over. So you'll notice on these two chromosomes, you have the genes for color vision, and they are supposed to line up correctly, right? So that if you have any crossovers, you would maintain all of those genes in all of the resulting chromosomes. However, because these genes are kind of similar in their sequence, sometimes a mistake gets made and you actually line up this green opsin gene with the red opsin gene on the homologous chromosome. If crossing over occurs, what you can end up with is a duplication and a deletion. So you have two separate products here. So this is actually how a lot of people become colorblind. They lose one of their genes due to a mistake in crossing over. Now, this duplication event is also a very important product for the species in general. And so if you duplicate a gene, what happens is both of those genes can now carry out the function of that green opsin gene. Normally, if you mutate a gene, you tend to mess with the function of that gene. If the gene can't do its function anymore, often the organism will show a deleterious phenotype. However, if you've duplicated your gene, what you have now is extra genetic material. So one of those genes may carry on doing that green opsin gene function, while the other one is now available to be changed and possibly to, over a long time period, to develop a new function. We will talk about gene duplication more when we talk about evolutionary genetics. There are a number of human disorders that result from deletions of partial regions of the chromosome. I'm going to tell you about two examples here. So one of them is Cri-du-Chat syndrome. This is caused by a deletion on the end of chromosome 5. These individuals have a pretty severe intellectual disability. Babies with Cri-du-Chat syndrome actually have a distinctive cry that sounds like a cat, hence the name of the disorder. Another human disorder resulting from a deletion is Williams syndrome. So Williams syndrome occurs on this chromosome where there's actually 17 genes between two genes that are very similar to each other. If you get an unequal crossing over, you can end up with either duplication or a deletion, and the deletion is actually Williams syndrome. Williams syndrome has pretty pleiotropic defects. There's cardiovascular issues, developmental delays, learning disabilities, that type of thing. So with chromosomal inversions, I want to make the point that there is no genetic material that is either gained or lost. And so this is simply a switching around of the order of the genes on a chromosome. So you would think that there wouldn't be a severe phenotype, and in fact, sometimes there really isn't a severe phenotype. However, occasionally you actually do get a phenotype. The reason why there sometimes is a phenotype is because genes are actually affected by their position on a chromosome. We'll go into this more when we talk about epigenetics. This example here is simply looking at an inversion that has taken place between two related species, humans and chimps. And you can see based on this chromosome banding pattern that even though these chromosomes are very similar otherwise, there's been this switch in this what's called a paracentric inversion of this region where the centromere lies. Next, I'd like to talk about translocations. So translocation is an exchange of a chromosomal region between two non-homologous chromosomes. This can happen in a couple of different ways. It can be a reciprocal translocation, so two chromosomes switching their arms, okay? Or it can be that one part of a chromosome gets tacked onto the end of a different chromosome. Here's an example of a reciprocal translocation where these two chromosomes switched their arms. This would be considered a balanced translocation as long as the individual had both of these parts. So in this case, there is no loss or gain of genetic material. You can imagine, however, that if you have an individual who has a balanced translocation, they will have trouble making gametes that are going to be viable. So here we have a normal red chromosome and a normal blue chromosome, and then the two that have the translocation. And this one is going to be normal because it has a balanced translocation, but in the process of forming gametes, if the gamete receives the two good chromosomes, that of course will be a viable gamete. Also, if the gamete receives the two with the balanced translocation, that will also be a viable gamete, and it can join with another normal gamete to produce a progeny. However, all of these other possibilities are actually unbalanced. That means that they are missing part of a chromosome or have extra of one of the parts of the chromosomes. And so here you can see if this full-size red chromosome joined together with this one in a gamete, you would have duplication of the red chromosome and you would be missing part of that blue chromosome, for example. It turns out that a lot of people actually have these chromosomal translocations, and this is a really big reason why certain individuals tend to have a lot of miscarriages. If a person has this type of balanced translocation, a lot of the conceptions that occur will be unbalanced and will be therefore lost in the early stages of pregnancy. Just want to tell you about one type of translocation that is pretty common. It's called the Robertsonian translocation. We've talked about Down syndrome and trisomy 21, and that is one way you can have Down syndrome. It turns out that there's another way that you can get Down syndrome that is due to a translocation. And so the Robertsonian translocation is actually an example where you have chromosome 14 is in red here, and chromosome 21 is actually physically attached to the end of one of the chromosome 21s. So what will happen here is each of these chromosomes will line up and segregate, and depending on which chromosomes end up together, you can end up with an individual with Down syndrome. You can also end up with a child who will be a carrier, again, for that translocation. But this individual will be phenotypically normal because they have the balanced version of the translocation. Finally, translocations have also been shown to be important in cancer in certain cases. There's two examples. One is a type of lymphoma, Burkitt lymphoma. If you get a translocation of chromosome 8 and chromosome 14, what you end up doing is you end up putting this MYC gene, which is a gene that promotes growth, next to the promoter for this Ig gene, which is going to be turned on a lot, right? And so this regulation means that the MYC gene is turned on all the time, and this cell will become a cancerous cell. Same kind of thing happens here, only it's a little bit different. The ABL gene and the BCR gene, they actually get translocated really commonly in myelogenous leukemia. This is a translocation between chromosome 9 and 22. This is actually a break in the middle of the genes so that the individual exons of this one gene get spliced with the exons of another gene. And this hybrid protein is actually an oncogene.